going down our list here. So now we said we saw the brainstem before. Now we can see the divisions of it a little bit easier here. So the bottom portion we have the medulla. So the medulla oblongata. So we have the spinal cord down here, and then meeting up to the medulla. Where spinal cord becomes medulla is a little bit blurry. There's like some decussation points or crossing over of some motor tracks, but those can be kind of hard to see. But
just a little bit bigger or something, or how do you identify it? Just more location, probably.
More location. There's a little bulge, but basically, closer to the brain is going to be medulla, and then later on it's going to be spinal cord. But again, a little bit.
And also, I've noticed a couple different ways of saying that: medulla oblongata on the right, medulla on the book.
Sometimes you hear medulla by itself or medulla oblongata, but it's referring to that first portion of the brainstem. And then next we have the pons, which is this like kind of thicker, bulgy spot, and that's going to be in line with the cerebellum.
So as far as function, like some of those like medulla and stuff like that?
Yeah, so the brainstem is one of those things where it doesn't have a nice, neat, like one, like temporal lobe we can give it hearing. It's like a nice, neat one little function. The medulla does a lot of stuff, or the brainstem does a lot of stuff, but in general, it's going to be like vital function. So here we'll have like respiratory centers to control breathing. We'll have cardiac centers to control heart rate, things like that. There's like some sleep stuff in here. So in general, we're thinking like vital functions. And I guess another thing, you'll see a lot of these nuclei for cranial nerves. So when I say nuclei, that in this sense, that's like the part of the tissue where these cell bodies are for all these neurons. And so you have like the origin of a lot of the cranial nerves here. And then actually, so clinically relevant, a lot of times you use like a neurologist will do a cranial nerve exam. So they'll check all these, different functions of the different cranial nerves, and that can help you localize pretty well where you might have, let's say if someone has a bleed somewhere here or a lesion of some sort, depending on what, functions are intact. If you can, lift up your eyebrows on one side, close your eyes, things like that, it can help localize all that. So that's the medulla, the pons, and then the midbrain here.
Actually, so the medulla and pons, you said all those different functions, it's all kind of together?
Yeah, so they're like a lot of these centers are kind of running up and down. Like the reticular formation is one that has like a lot to do with sleep that kind of runs along the whole length of it. But a lot of the cranial nerve nuclei are just kind of scattered throughout that. So that's the brainstem. So medulla, pons, and the midbrain, and the spinal cord would be back there. So let's see what.
Let's just go up to the top here. Maybe kind of work our way this way, maybe.
Yeah. So the cerebellum has to do a lot with balance and coordination. So it's going to be working with. So we said before that primary motor cortex of the frontal lobe, right at the back of it, was muscle control, and that's going to be doing a lot of talking with cerebellum for coordination of that.
Okay.
So if you have damage to the cerebellum, that's where you start seeing like ataxia, which is like uncoordinated movement, things like that. And one thing we'll mention here, so we mentioned the arbor vitae before, so that literally means just like tree of life. So that gives us a good, we can see really, really well the difference between white matter and gray matter. So
what's the slight difference between those two, white and gray matter?
So white matter is referring to neurons that have a myelin sheath around them. So that's essentially like a lipid bilayer or like fat that's wrapped around it. So that's like insulation. So if you have like more or less, you can think of neurons like wires carrying electrical information. And so wires that are insulated are going to have information travel faster than if they're uninsulated. And so that's different. So white matter is going to be insulated, whereas the gray matter is going to have unmyelinated. Okay, so maybe next we might look at the ventricles.
Okay.
So we had mentioned before the cerebrospinal fluid, the CSF, in the subarachnoid space below the arachnoid mater. So the ventricles are they're connected to, they're continuous with that subarachnoid space, and in the ventricles, it's like essentially these little like holding spots for CSF within the brain, and that's also where CSF is produced. So there's these cellular structures called the choroid plexus. You can't really see them on a gross dissection like this per se, but that's going to be where the CSF is produced. And so the CSF in general is just this fluid that the brain is encased in because it's all the way around brain and spinal cord, and it has multiple functions. There's a little bit of a nutritive function, so it brings some nutrients, but probably the most prominent function that's been identified is that it is protective. So the brain is kind of like sitting in this cerebrospinal fluid, so it helps with, if you get hit in the head, it's a little bit of a protection against like hitting against the skull on the other side. So there's 4 ventricles, and so 2 lateral, a third, and a fourth ventricle is how they've been named. So we'll start up here. This is a good example. We have the septum pellucidum here. So this is the separation between the 2 lateral ventricles. And actually, I might puncture through this one just so we can kind of see. So when you cut the brain yourself, only one side will probably have that, because it's a little membrane right in the middle. So if you're on one side of it versus the other. But so this is the separation between the lateral ventricles on either side. So essentially, all the ventricles are then is just these little spaces that are filled with CSF. So it's like right behind the septum pellucidum here. So lateral ventricles here are going to be like, so other big things you'll see is the corpus callosum around lateral ventricles.
So the ventricle is just basically a space.
Yeah, the ventricles are just a space.
Okay.
A space that are full of cerebrospinal fluid in an intact organism. So then a little ways back, we'll have the third ventricle. So third ventricle is going to be in proximity to the thalamus. So we'll do these other structures in more detail in a second here. But third ventricle here, thalamus, and then so the lateral ventricles are connected to the third ventricle. So you have,
this is 1 and 2 up here, and then 3.
Yep. So lateral ventricles, which would technically be ventricles 1 and 2.
Yep.
Third ventricle, and then that flows down further here to the fourth ventricle, which you see in this space down here between the pons and cerebellum.
Okay.
And I guess so like a clinically relevant thing with that is that, let's say you have some sort of a blockage somewhere, maybe there's a tumor or something that is blocking this flow of fluid, you then could get a buildup of pressure. So let's say like we have a block here somewhere, the lateral ventricles could build up in pressure because it can't flow out like it's supposed to. And then that can cause some problems with, increased intracranial pressure and things like that.
Okay.
Okay, so now we talked about those. Let's we had mentioned the corpus callosum before, so that's this band right here of white matter.
What's the function there?
So that is the communication point between the different hemispheres. So this is how information from the left side of the brain gets to the right side of the brain and vice versa. And so we had mentioned with that activity. So this is where that kind of comes into play with the visual stuff. So if you in very severe seizure disorders, sometimes, very rare, this isn't because we have a lot of good medications now. But in very, very severe refractory seizures, you might see a surgical procedure to kind of cut this corpus callosum. So a seizure essentially is just the brain is like firing way too much. There's way too much electrical activity. So you got to calm that down a little bit sometimes by cutting some of this communication point. But then one of the weird things you'll see with that is that the right side of the brain can't communicate as well with the left side. So you start seeing some like very weird very specific symptoms where let's say you have something in your right or your left visual field that's getting the right side of your brain, but then if it can't get to the language center, you maybe can't say it, even though you know what it is. But that activity talks more about that, about that stuff.
Absolutely.
So that's the corpus callosum. There's some other structures here that would be associated with hypothalamus or with the hippocampus. Which hippocampal formation we don't have on here because it's kind of hard to see. Part of it is the fornix here that would connect to that, but it's kind of a 3-dimensional structure, so it's really hard to see when you cut right down the center of it because part of it's going to be kind of extending out of our plane of cut here. Then we have the thalamus here. So the thalamus is kind of like a relay center, a relay station. So we have a lot of information coming up from the spinal cord into the thalamus, then it's going to different areas in the brain. So this is kind of where we have information. It's deciding where information is going in different parts of the brain. And then below that, we have the hypothalamus region here. So, and hypo just means below. So hypothalamus, that does a lot of, there's a lot of endocrine or hormonal control there. So, and there's like a thirst center and some other things there. So, this is actually another thing we're going to mention with some of these specimens. So this specimen here is nice. We have the pituitary gland intact, and that may or may not be intact in your specimen.
Yep.
So pituitary gland, another name for it is hypophysis. You'll maybe see that I think we saw it on the website.
Yep.
Hypophysis intact. So that's our name, pituitary gland. So the hypothalamus here is going to communicate with the pituitary gland, and then there's going to be a lot of different hormones that are released into the blood from pituitary gland here. And one thing that is relevant, so we also saw here this optic chiasm. So this we can see here. So this is where the optic nerve would be coming out of the plane here into here, and then it's crossing over, and then this is where it's coming from the other side. Back over here. So one thing that's really relevant is that a lot of times you'll see with like a pituitary tumor, for one, you'll have a lot of hormonal imbalance because that's releasing, that's like a very, very important hormone releasing center. But you might also see some visual issues. So you can imagine if there's a tumor here that's compressing up against the optic chiasm, you'll have very characteristic visual disturbances. So a lot, the reason, and that actually brings up a good point, why it's so important, especially if you're thinking of going into healthcare in any capacity. Learning the anatomy is important because then when you know structures that are close to each other, you can see how certain problems would manifest.
The pineal body, maybe we talk about that one at all?
Yeah. So the pineal body or pineal gland is then going to be kind of posterior to the thalamus section. So the pineal body or pineal gland is responsible for releasing melatonin. So melatonin, we think of, you might take a melatonin supplement. People take those for jet lag sometimes. And so this is going to be involved in our circadian rhythm, like sleep cycle. So you actually have some visual information that will come directly from the optic nerves and feed into that. So that'll help kind of entrain, when it's night and day by like seeing light and dark. So
maybe it mentions what this kind of see the cerebrum here too. We didn't really mention that. We talk about it on the outside, but not necessarily inside though. So
yeah. So this again, we can see all this folding of the cerebrum. So there's lots of different, all these gyri, they have different names. At this level, that's it can get overwhelming really quickly. So here we can see how all this folding of the brain tissue up here allows for a lot more increased surface area to fit in a smaller structure. Otherwise, the skull would have to be so big if we didn't have all this folding.
Kind of wondering too, do we want to maybe do a cross section at some point? Maybe show that white and gray matter?
Yeah, we can.
Or anything else you want to identify here? I think that might be most.
I think we got most of the structures that we had wanted to talk about. Again, there's a lot of stuff. So like sometimes you'll see in references, like there's a lot more structures in here. It can get overwhelming pretty quickly. But these are like some of the big ones that we had pointed out for this. So
absolutely. You want me to try this one here with that cross section? See if we can see some of that white and gray matter in that main area there.
Yeah, and then you can also so we did like a mid sagittal section where we cut down the along through the center of this longitudinally. You can also do some of these like coronal sections where you're cutting this way, because then there's a couple other structures you can see, but they're really hard to get the section in the right spot, and a lot of those structures. So for example, to see the hippocampus or the amygdala, you have to kind of have your coronal section cut like perfectly, which is pretty hard to do. So if you're interested in those, like that can be done, but it's quite a bit harder. Okay. So but yeah, we'll try a.
Just gonna show it here for the camera there.
Yeah, we'll do a. And what I'm looking for here is I'm just kind of lining it up for the temporal lobe. That's usually a place where you get a lot of these coronal sections. And actually, I'm gonna use the scalpel for this because we still have a lot of dura mater intact here. Here we're just gonna try to see the difference in white matter versus gray matter. So you can see it's very hard to cut through the dura mater with just your plastic knife, obviously.
Oh, yeah.
Okay, so it's.
And then show up the best. Should one more cut, maybe?
Yeah, so we're looking for the difference between white and gray matter. The problem with these is that since we just did a fresh cut, it can be a little bit hard to see the contrast.
Try to put more here. I would try this one here.
You can kind of see white and gray, but we'll try to get a better view here.
This one's cut right here. How's that
right
there?
This one, yeah. Oh, that one's really good right there. So
actually, I can get both sides here.
So now we can really like a lot easier see the difference between the white matter here. Gray matter is what we talked about. This is myelinated axons that are insulated. This gray matter is unmyelinated. And then, so we didn't, it's a lot harder, like we said before, to see the structures when you're in these coronal sections. One, because it's really hard to know what level you're at, anterior to posterior. But it can be interesting, looking at these. This actually shows the corpus callosum really well. So we can see, like, let's see, is this the front or? So this would be the back. So this is the right hemisphere, left hemisphere. You can see how this corpus callosum, so if we show this right here. So this was corpus callosum right here. You can imagine if you do a coronal section like that, that's what we're seeing here. And then you can see how this is allowing the right side to talk to the left side by that tract running across like that.
Absolutely. Anything else you can think of that we missed?
I think we had hit most of the ones we had wanted to look at. Again, there's a lot more, but it gets overwhelming really quickly.
Thank you so much, Blaise. Like I said, there'll be a check sheet and extra materials available on the website. Thanks again.